screen visible to you?
Yes, sir.
Okay. So in the last class, we discussed about different form of nitrogen content which might be present, and depending upon the different stages of decomposition, what are the available nitrogen form which we can get. Now the next immediate one which we need to consider is other metals and chemical substances which are significant, and the effect is also significant as far as public health is concerned. So if we see here, so several types of apart from whatever we have discussed, several other metals and chemical substances like iron, manganese, copper, lead, barium, cadmium, selenium, fluoride, arsenic. These are the metal which can be found in the natural water, and mm, they can be present in the water, and the amount of these minerals or metals in potable waters are then limited to their permissible standard values, so that the harmful effects to human life can be avoided. Right? So the type of harmful effects that are caused by various elements and compounds, I'll be discussing in subsequent slides. But for example, if you see the concentration of iron and manganese is not allowable when it is more than zero point 0.3 ppm. What happens is when we have excess excess of these metals, this will cause discoloration of the clothes during washing, and as well as incrustation in the water mains due to the deposition of the ferric hydroxide and manganese oxide. And somehow you can relate this with the deposition within the boiler also. So lead and barium are very toxic. Most of you know that, and very low ppm of these are allowed. So otherwise the entire water will be called as toxic. Arsenic and selenium are also very poisonous, and arsenic pollution, how detrimental it is as far as human health is concerned, and it can cause severe permanent damage to the health of the human being, and it may cost totally, or therefore they must be removed totally. So you have to ensure that the water which you are supplying to the water supply scheme are not having any trace of arsenic or selenium. Human beings are affected by the presence of high quality of copper in the water, and that is why it is also a serious matter and to be taken care of properly. Fewer cavities in teeth will be formed due to excessive presence of fluoride in water, and the maximum limit is 1 ppm. So fluoride has very negative impact as far as teeth is concerned, and a laxative effect is caused in the human body due to excessive presence of sulfate in water. So these are the different type of problems which we can encounter, and if we have water whose quality is not good. Fine. So that is why we have to be very careful about the quality of water, and what are the metals that particular water does contain, and what are the other chemical substances, and how to take care of them, and how to lower down their limit. That is also a very important part to be considered. So the amount of most of the metals and the other substances present in the water can be estimated by using color matching methods, by using different indicators. So while talking about nitrates and nitrites, also we talked about colorimetry with great extent, and that is why I am not going to discuss them again. However, what you need to remember is all these different chemical substances can be estimated by the color matching techniques, but for each case we'll have different indicators. Fine. So, as I was talking about the health effects of chemical parameters, and in these particular tables, I tried to give for the most common metals and chemical substances and their maximum limit as per BIS guidelines, and their general health effect. So here, calcium, magnesium, iron, manganese, aluminum, copper, all are given, and you can see like they can cause serious damage. Like copper can cause liver damage, mucosal irritations, renal damage and depression. So uh, they are severe. Neurological disorders it can cause. On the other hand, several other problem, not only related to human health, but also it's related to the other uh, properties which will be used for some other public interest, like pipes, boilers. So those are the components which will also be affected if we don't maintain the quality of water within the maximum allowable limit. Now, similarly, we can see here the effect of zinc, ammonia, nitrite, nitrate, sulfate, chloride, and fluoride. You can go through them, and for your better understanding, I have given this, and you can go through them, and you can see that general and health effect of all these parameters and why they are so significant as far as quality of water is concerned, and as far as human health is concerned. So that is why once you see one particular parameters and how it is going to affect, if the concentration is more than the allowable, then it will give you a very good idea about how dangerous that particular element can be. And then we have phosphate, arsenic. So arsenic, though we have given a maximum allowable limit, we try to keep it as less as possible, and it is best to provide the no arsenic stress. Similarly for mercury, it is also highly toxic, and it causes a very infamous Minamata disease. So neurological problem it creates. So it's a heavy metal. Cadmium is another one heavy metal, highly toxic. Lead is another one. Chromium and pesticides. So in this case, if you see that the limits are almost zero, we can consider. So since the toxicity of the substances are increasing, the allowable limit is decreasing in this case. Now, next part which we will be discussing now is dissolved oxygen, because chemical substances, you can go through this table and you can have a better understanding about Am I audible now?
Yes, sir. Yes.
So up to how much you could hear? Please tell me the exact point up to which you could hear. So I'll start from there. Those who are listening, please tell me up to how much could you hear? Hello?
Yes. The
Hello?
last slide, sir.
This
Just
one. This one, dissolved gas.
yes, sir.
Okay, so you could hear about dissolved gas. So the next one is biochemical oxygen demand. So that is what I need to repeat again, right?
Yes, sir.
So dissolved gas is fine to all of you?
Yes, sir.
Fine. So be prompt in responding. I have been telling you, all of you, for so many days. So, biochemical oxygen demand is the most important parameter which should be studied and which gives us the idea about how much decomposable organic matter present in the water. Right? So, this is basically a reflection by which we can understand the overall quantity of organic matter which are present in the water that can be decomposed by the help of oxygen. Right? So, that is why we say here that the extent of organic matter present in water, and that is the way we basically define it. So the extent of organic matter present in water can be easily estimated by supplying oxygen to these samples and finding the oxygen consumed by the organic matter present in the water to decompose, to get decomposed. Now this oxygen demand is known as biochemical oxygen demand. Now if the water is contaminated with sewage, the demand of oxygen by organic matter in sewage is also known as biochemical oxygen demand. So this is a situation, if such kind of contamination happens in the water, that would surge, that would surge the overall biochemical oxygen demand or overall oxygen demand to decompose the organic matter. Just 1 second. Fine. So hope you have understood. Are you able to hear me?
Yes, sir.
Okay. So if you see the overall oxidation, the overall process of decomposition, that would give you a better idea about how this entire process happens. So uh, any sort of organic matter present in the water, first aerobic action would start. Because of that, you have some dissolved oxygen present in the water itself. Now, because of that, the organic matter would start decomposing, and it will continue until and unless the dissolved oxygen is exhausted completely. Now, once the dissolved oxygen is exhausted completely, the anaerobic action begins to happen, and since that point onwards, the foul smell would get emitted from that particular water bodies, because once you have anaerobic decomposition started, you will have methane, carbon dioxide, those gases which would emit, and because of that, obviously there will be a stinking effect, and that emit the foul smell. Now this aerobic decomposition of organic matter, again it happens by two different stages which we are going to discuss now. So the first one is the carbonaceous stage, right, or carbonaceous decomposition of the carbonaceous matters. So the carbonaceous matters are the first oxidized, and the oxidation of nitrogenous matters takes place later stage once the carbonaceous matters are decomposed. After that, the oxidation of nitrogenous metals starts happening. So the biochemical oxygen demand can again be defined in a from a different perspective. So the biochemical oxygen demand is an empirical standardized laboratory test which measures the oxygen required for aerobic oxidation of decomposable organic matter and certain inorganic materials in water, polluted waters, and wastewaters under controlled conditions of temperature and incubation period, which is also very, very important to remember. So, this definition is from the perspective of laboratory test. We provide ambience for the organic matter or uh, basically microorganisms to grow, and that is the way this test is performed in a certain temperature, in a certain environmental factors, and this particular test is done. So the quantity of oxygen required for above oxidation process is measured for this particular test. So dissolved oxygen in the initial stage and dissolved oxygen after the decomposition or after a certain period, and the difference between them would give us the BOD value. So that is the way we determine BOD in the laboratory. So this test, is applied for fresh water source as well as wastewater, like any sort of water which is contaminated or natural water where you have organic matter. You can perform this test in order to determine the BOD value, and that is an indicator of how much organic matter present in this particular water. Fine. So the next is we are going to talk about just 1 second. Yeah. Yeah. So screen is visible, right?
Yes.
Yeah. So now we are going to talk about the methodology. So the methodology of BOD test is to compute the difference between the initial and final dissolved oxygen of the samples which will actually keep in the incubation. So what we need is we need a minimum of 1.5 liter of sample for this particular test, and DO dissolved oxygen is estimated by iodometric titration. So this is a one type of titration. All of you would perform this test in your environmental engineering laboratory. You would have a better understanding how you are doing that. Now, since the test is bio-assisted procedure, so it is necessary to provide standard condition or ambient conditions as in terms of nutrient, in terms of pH, in terms of adequate population, in terms of temperature. So these are the different factors or parameters should be provided to perform this test, because this is a bio-assisted test. We want the microbial growth to happen, and we'll have to ensure any presence of microbial growth inhibiting substances in that particular water in which we are going to do that particular test. So the low solubility of oxygen in water necessitates strong wastes to be diluted to ensure that the demand does not increase than the available oxygen. Then it will be again a problem. Once you start doing this test, you have to perform the entire test within the aerobic zone. So if your contamination is very high, what you do is you first dilute the sample to ensure that during the entire procedure of the test, all the organic matter will be decomposed, and after that also there will be a certain amount of oxygen which will be present. So before complete decomposition, the exhaustion of the dissolved oxygen is not desired. That is what you have to keep in your mind. Okay, fine. So after that, what happens is a mixed group of microorganisms should be present in the sample. Otherwise, the sample has to be seeded. So that is done artificially by adopting some microorganisms. Generally, the temperature is controlled at 20 degrees centigrade, and the test is conducted for 5 days. So universally, this is what is actually fixed. So if you do 5 days, as 70 to 80% of the carbonaceous wastes are oxidized during the first 5 days. So the test can be performed at any other temperature provided, and then you need to conduct temperature corrections, and again you have to express that particular result in terms of BOD5 at 20 degrees centigrade, and that is the universally accepted one. So even if you perform in some other temperature, and if you perform the time is also more, so ultimately you will have to express that in BOD5 value. Fine. So while reporting the results, the incubation period in days and the temperature in degrees centigrade is essential to be mentioned, obviously. And the BOD5 is basically BOD of 5 days equals to loss of oxygen in milligram per liter into dilution factor or ratio, whatever you say. And BOD of safe drinking water must be nil. So there should not be any organic matter present in the water, especially for drinking water. So now we'll be talking about, so the test procedure is there is an elaborated test procedure. I am not going to discuss over here. So you will be performing the test in the laboratory, and through the, once you perform, you will have the entire idea. So I just told you that there will be iodometric titration. The rest of the process is whatever we have discussed. Accordingly, we will have to do it. So now we'll be talking about bacterial and microscopic characteristics. So this is also very, very important, bacteriological and microbial characteristics, because whatever we can't get in the organic matter, here we can see them, like each one bacteria can be recognized by this process. So the examination of water for the presence of bacteria is important for the water supply engineer from the point of public health aspect. Now the bacteria may be harmless to the mankind or may be harmful, but we should be able to recognize them. So if the bacteria is harmless, then we say that the particular bacteria is known as non-pathogenic bacteria. So most bacteria are harmless and under certain conditions beneficial to human beings and animals and other crops. and that is known as non-pathogenic bacteria. There are certain bacteria, they are deadly for the man and as well as the animal. So those are known as pathogenic bacteria. So we'll have to be very, very careful, and we'll have to ensure that no pathogenic bacteria are present in the drinking water. Some of these are able to multiply and continue their existence while the remaining die out in due course of time. So that's a general features, and many of the bacteria found in water are derived from air, soil, or vegetation. So based on the requirement of oxygen, we can again categorize this bacteria. One is aerobic bacteria, another one is anaerobic bacteria, another one is facultative bacteria. So if you see, aerobic bacteria are those which require oxygen to survive, or their survive. Anaerobic bacteria is those bacteria which does not require oxygen, or free oxygen for their survive. So they can survive, in absence of oxygen, free oxygen also. And facultative, those which can survive with or without oxygen, these are known as facultative bacteria. So, and now another one important part is, pathogens we have discussed, non-pathogens we have discussed. So, depending upon the size or the shape and size, we have several type of characterizations of bacteria. Cocci, streptococci, staphylococci. So we have bacilli, we have vibrio, we have spirilla. So different type of bacteria are there depending upon several characteristics. We are not going to go in those details. One important part here is the what is coliform. So that is very important, because this term will be using several times in the due course of time, while talking about the bacteriological and microbial characteristics or microscopical characteristics. So let us define the coliforms. The coliforms are the rod-shaped or non-pathogenic bacteria, mostly bacilli. So bacilli, you will see they are rod-shaped. Fine. So the coliforms are rod-shaped, non-pathogenic bacteria whose presence or absence in water indicates presence or absence of fecal pollution, and hence of the pathogens. So though they are not pathogenic bacteria, but their presence would create pathogen to come, and that is why their presence or absence in water indicates presence or absence of any fecal pollution, or any pathogenic bacteria to be coming to that particular water. So the selective medium that promote the growth of the
Sir, excuse me, sir.
Yes.
Sir, what is facultative bacteria? The third one.
Yeah, third one, it can survive in presence as well as in absence of oxygen.
Okay,
Understood? Sir. In both the situation, it can survive. Understood?
Yes, sir.
So that is the facultative one. So any sort of chance of fecal pollution can be understood from the coliforms, the presence of coliforms. Fine. The coliform group, fecal and total, are the most widely used indicator organisms. And the total coliform group consists of members whose normal habitat is the colon. Fine. So lower portion of the intestine of human, and worm and other cold-blooded animals. You can see some members of this coliform bacteria present in soil and as well as in the vegetations phases also, but mostly they remain in our intestines, and not only us human beings but also any other warm or cold-blooded animals. Now, among these coliform, which are exclusively seen in fishes and which are not found in the soil or vegetation, that is basically the 96% of the total coliforms. Fine. So those coliforms are known as fecal coliforms, and they have also recently been named as thermotolerant coliforms. Fine. So fecal coliforms is another one term which we need to remember. For bacteriological analysis, the following tests are done. So now we'll be seeing what are the different tests are there to understand the different type of bacteria and their quantity and all those things. So the first is standard plate count test or plate count test. So in this method, total number of bacteria present in a milliliter of water is counted. Right? 1 ml of sample water is diluted in 99 ml of sterilized water or deionized water. So the water is absolutely sterilized, and we can ensure that no organic matter and chemical substances present in the particular water. So sterilized water to dilute, and 1 ml of dilute water is mixed with 10 ml of agar of the gelatin. This mixture is then kept in the incubator for 37 degrees centigrade for 24 hours or 20 degrees centigrade for 48 hours, depending upon how much ambient temperature you get or is available in a certain area where you are performing that test. So after the sample will be taken out from that incubator, and the colonies of bacteria are counted by means of microscope. So in the microscope you will see that colorful bacteriological colony which are formed because of this growth. So drinking water should not have more than 10 coliforms per 100 ml of water, which we are planning to distribute in the distribution system. Next is MPN, multiple tube fermentation technique for coliform bacteria. So MPN is really important, I'd say, and it's the most widely performed test, most probable number. That is why we say MPN test. So first, let us define most probable number is a number which represents the bacterial density which is most likely to be present. Fine. So most likely to be present, and the bacteriological density. So most probable number is a number which represents the bacteriological density. So after formation of colony, whatever dense bacteriological bacteriological density you get can be called as most probable number. So what is done is in this test, this is actually done by multiple tube method. So in multiple tube method, a series of tubes containing a suitable selective broth culture medium. So this broth culture, there are several broth culture medium which are commercially available, and bioengineers or bioscientists, they also can create such kind of broths, and is inoculated with the test portions of the water sample. Now, after a specified incubation time at a given temperature, each tube showing gas formation is regarded as presumptive positive, since the gas indicates the possible presence of the coliform. Actually, I'll stop here today because I'll have to go for a meeting. I forgot that. Just wait.